Okay, so let's see if we can do this. Talking about chapter 16, the nervous tissue. I did my best to draw some of the stuff up here. It's rough. Bear with me. Hopefully, I'll be able to go through some more of the information than what we were able to talk to in class and have you understand it just a little bit better. Kind of do it at your own pace. Anyway, so remember that when we talked about the nervous system in class, I said that I want you to break it down into two parts initially. And we're going to talk about the central nervous system, and we're going to talk about the peripheral nervous system, so CNS, PNS. And for the CNS, we say that is the brain and the spinal cord. And then for the peripheral, we just say like, well, that's everything else. Now the reason that we're just kind of lumping that as everything else is because chapter 17 and 18 are going to talk about the brain and the spinal cord in more detail. And then we're not going to end up talking anymore about the peripheral nervous system after that. So we're just going to kind of lump all that together for a second. In the future, you'll learn more about it, obviously. But the next thing that you want to learn about, and we talked about this when we were looking at tissues in chapter 5, is that there are two kinds of nervous tissue. Number one are neurons. Number two are the neuroglia. Now, one of the facts that you need to know about that is that neuroglia, especially according to your notes, outnumber the neurons like 20 to 1. You have an incredible amount of neuroglia that are there to help support and provide the overall functioning of the neurons. So the 6 types of neuroglia that you need to know are 4 that are related to the CNS and 2 that are related to the PNS. Astrocytes, oligodendrocytes, microglia, and the epidermal cells. All of those are central nervous system specific. The other 2, Schwann cells and satellite cells, are going to be specific to the peripheral nervous system. Now, 2 that have the same job, but in different areas, oligodendrocytes and Schwann cells. They both provide the process of myelination. And what did we say? That is a production of myelin. Myelin is that fatty protein-like substance that goes around an axon and allows it to speed up the impulses or the transmission of information, the transmission of energy, essentially, down the axon. We'll talk more about that here in a minute. But even though they're located in 2 different areas, they have that same job. You want to know that. With neurons, they don't ask you much more about neurons, other than being able to label it, and we'll talk about that. But you have different kinds of neurons. You have unipolar, you have bipolar, you have multipolar neurons, and they all do slightly different things. You'll find them in different areas of the body. Some of them are going to be specific to particular senses, and then others are going to be very specific to, for example, the innervation of muscle cells. We'll talk more about that. But you want to know some of the jobs that are related to the CNS and to the PNS. For the CNS, we have things such as your senses, the sense of sight, smell, taste, feeling, hearing. All of those are going to be very CNS based. Know that. I'm getting in the way of myself with this. Let's take a look at my crude drawing from the book. The book, or the notes, or your manual, show you 3 different pictures. One picture is an electron microscope picture. Another one is, I'm blanking on the SEM picture, and then they have the drawn picture. I did my best to recreate that one as we're going through this, because that's what you're going to see on your quiz. That's what I'm going to ask you to label. When you're looking at this, regardless of what type of neuron we're talking about, again, unipolar, bipolar, multipolar, they're all going to have 3 things in common. You're always going to have an axon, you're always going to have dendrites, and you will always have the soma, or the cell body. Always. Doesn't matter. In this case, this area right here, that's your soma, that's your cell body. Now there are things within that we're going to talk about. We're going to go through this as the process occurs. And the process being the process of receiving information via neurotransmitters, creating this wave of depolarization, which then creates an action potential. I'm getting ahead of myself. These areas right here, around the edge, those are your dendrites. Those are like the antenna that receive information. And now instead of receiving information in the form of like a radio signal, they receive information in the form of neurotransmitters from other neurons. Where does that happen? Right here. This little area is the terminal of another neuron. This is called your presynaptic junction. This presynaptic area has a little gap right here that we're going to talk more about, and that's called your synaptic cleft. It releases neurotransmitters, it releases information that then is picked up by the dendrites and sent down through the soma to this guy right here, who is called your axon hillock. The other name for this that I want you to know is trigger zone. Now before we get ahead of ourselves, before I get ahead of myself again, we're going to scoot back and we're going to talk about the soma for just a second. This right here, like every other cell that we've ever talked about, that's your nucleus, that's your control center. This area around it, though, is familiar with what we've talked about, but it's slightly different. The neuron has its own take on it. These are rough endoplasmic reticulum, just like we've seen in the cell when we talked about it in chapter 3. But instead, they have these blue-bodied RER that are called the blue Nissl bodies. And they still synthesize, they produce proteins, but instead of thinking of them as just proteins, now we're thinking of those proteins as neurotransmitters. So it's going to synthesize and produce proteins in the form of neurotransmitters that are then going to be available for release from the junction. That dendrite, or the wave of depolarization that came in through the dendrites comes down to your axon hillock. That trigger zone gets its name because if it hits a certain point, it triggers an action potential. And until it hits that mark, action potential doesn't occur. It is up to that to have it occur so that it sends that action potential down the axon, down to the terminal junctions. Now, this is your axon right here. This is the area along which energy or information or that action potential is going to be sent. But on either side of it, you can see, now your book has it as a better color. It does it in yellow, which is a little bit better, in my opinion, as a representation of that fatty protein substance, which is going to be myelin. I didn't have yellow, so I went with what I had, which is green. That lump right there that coats it, that myelin, allows it to speed up information being sent down. And along it, right in there, is the cell that's responsible for making that happen. Right there, that's the nucleus of your Schwann cell, a little black dot, that is creating that myelin that is surrounding it. It comes down, it has a little bit of a gap, and it starts up again right here, and this can go on and on and on and on. I only drew it 1.5 times. But that gap right there, that gap is important. It's called your node of Ranvier. It doesn't have any myelin. As this comes down to the end, this area right here, these are your terminal junctions. What does terminal mean? It means end. Now, an axon, or I'm sorry, a neuron could have one axon, but at the end of it, even though it only has that one axon, it could have dozens or hundreds or like 1,000 terminals at the end of it. And those terminals each can do something called innervation. And if you look at that word, innervation, it is a process by which a nerve is going to be going into a particular cell. So these notes are looking at motor units, because we just talked about muscles for the entire body. When a neuron innervates with a muscle cell, we call that a motor unit. Now, motor units have a wide-ranging level of innervation. Your notes talk about that you could have something as few as, say, 1 neuron for, say, 5 muscle cells in the eye, and that gives us incredibly precise control. And that high degree of precision allows us to control the eye in very subtle ways, to look at things, to be able to have a lot of control, what's called fine motor control in that area. And then we take it with something, their example is to talk about the gastrocnemius or the calves, where you could have just hundreds and hundreds and hundreds, and like all of this innervation. One neuron could be innervating all of these different muscle cells. And what did that do? Well, when one fires, all those muscle cells fire, and it creates strength because it creates an opportunity for the magnitude of that contraction. So the way you want to think about it is the fewer motor units, or the fewer muscle cells that are innervated by a single axon, the higher level of precision that they have, but often the smaller amount of strength that they have, the magnitude of the contraction. If they have more muscle cells per axon or per neuron, then you're going to have a higher magnitude or a higher strength within their contraction, within their activation. When we look at the terminal over here, there's this area right here. Now, what I did is, like that right there is supposed to be the dendrite of the next axon. And along that dendrite, there are areas that are enzymes and they're receptors. What happens is you have this gate. Now, you don't need to know this for the quiz, but just to help it kind of make sense of what you're looking at and why you're looking at it. You have this voltage-gated channel, and calcium goes in there. Now, when the calcium ion goes in there, it activates it. It's a turn-on signal. Flip the light switch. And once that happens, these are called vesicles. Now, I tried to, again, this is even worse than this over here, but this is a depiction of what's in your notes for that synapse. These vesicles contain the neurotransmitters that were created up here. When that gate is triggered by calcium, they go through these ends here in a process called exocytosis. And that word, when you break it down, exo, outside, cyto, cell, the process of taking a cell outside. That cell comes out, and those neurotransmitters will bind, if there's room for them, on those receptors or on the enzymes. The ones that don't end up coming back up, and they get recycled in a process that's called reuptake. So they are encapsulated in the vesicles, they get released through the process of exocytosis, they come back up, boom. Now these other structures in here that you'll want to be able to label but don't need to worry about too much, you have the neurotubules. They create kind of like an exoskeleton for the framework of the synapse, and this is your mitochondria. But the ones that I want you to actually understand what's happening to a certain extent so that you know what you're looking at and have some association with it are the process of the vesicles going through exocytosis and coming back up. Okay, so for the quiz, what you definitely want to make sure that you know and understand are going to be the breakdown of the nervous system, like what is involved in it, brain and spinal cord for the CNS, the two types of nervous tissue, neurons, neuroglia, what are the 6 kinds of neuroglia, the 2 that have very specific jobs, how are they broken down between the CNS and the PNS, what is that process called, myelination? Don't say that it is a process of creating myelin. Don't use a word to define a word. Yes, you are creating myelin, but what is that? It is the process of creating this fatty substance that then coats the axon and does what? It speeds up impulses. That's what I want to hear when you're going through it. And that myelination is done by the oligodendrocytes and the Schwann cells. When we come over here, I need you to be able to label this picture. I need you to be able to label this picture, and then understand certain concepts such as action potential, which is going to occur here at the axon hillock, motor unit, and the idea that innervation is the process of having a neuron combining with a muscle cell. If you have less muscle cells per neuron, you have higher levels of precision. If you have more muscle cells per neuron, you have higher levels of strength that go with it, or just the magnitude of the contraction once they're activated. In this idea that you could have a single axon, but then end up having dozens or hundreds or up to like 1,000 terminals per that single axon. Okay. I think that covers everything that we want to talk about. Let's move on.